Welcome you all to this Cryo-EM course. Last class we discussed about how to do carbon coating, as well as I discussed how we can use the carbon coater to coat the grid, and what is the mechanism. And then we also discuss about we have to do the glow discharge before we make our any TEM or cryo-EM sample. And glow discharge is very important, because if we do not do glow discharge, or if we do not prepare our grid properly, most of the time sample will go away, your biological macromolecule will not attached properly or will not fixed on top of the grid. So, it will be very difficult to see your sample properly, and also your grid will not absorb the staining solution properly. So, your staining will be very uneven. Now, we are going to see how we can do the negative staining, how we can prepare the negative staining grid. So, here today's topic is that we are going to discuss about the negative staining sample preparation. So, first let us take a look what is the requirement for negative staining grid preparation. So, here you can see this is a grid box. I already discussed about this one. The grid box, it has total 50 or 100, you can keep it in both the sides. So, this side 50 and another side there is another 50 grids, you can keep it. So, you need a Hartmann filter paper. This filter paper is also very important, because you have to blot your excess buffer solution, you have to blot your excess staining solution, and then next your sample it will be ready. You have to take this filter paper and then cut it in a triangular shape and keep those things, because the small triangular shape filter paper, it will help us to blot our sample. Now, our grid is here, we take this grid, we do the glow discharge, and then freshly glow discharge grid we are always using for TEM sample preparation or cryo-EM sample preparation. So, it is not a good idea to glow discharge your grid one day before or 2 days before, and then you use it for sample preparation. So, it will be always good to glow discharge, and immediately within 2-3 hours you use these grids for your sample preparation. And here you can see different types of tweezers are here. These are the tweezers. So, there are some curved tweezers are there, and then this is the empty grid box. Generally we are using this empty grid box to coat with plastic coating and then carbon coating, and then we can use it. That already I gave you all these examples, so I am not going to talk about all of these things. Now, you have a sample here that you can use it. You need a pipette. So, generally 2-3 different pipettes are very important, because we need that pipette to take the sample. Generally for negative staining we are using 3.5 microliter samples. Always remember that for negative staining, as I mentioned, that for cryo-EM, if we just consider cryo-EM 1 to 2 mg per ml. So, ml samples are required. Where the negative staining TEM, you need 10 times lower concentration. So, even if you have this concentration, you have to dilute these things 10 times or 20 times, and generally 0.05 to 0.1 mg per ml is a good solution. But always remember that this concentration, whatever I am talking, it may be a protein sample and it may vary based on their protein size. So, the same concentration, if you try a lipid protein complex, it will be completely different, or virus particle, because size is increasing drastically. So, generally a 300 kilodalton protein, if you keep this kind of concentration, it will be good enough for and it will be well distributed on top of your grid. So, generally for negative staining TEM we are using this concentration. So, we we need a pipette for sample, then we need another pipette, we have to take out the staining solution. Generally, as I mentioned, we are using heavy metal staining solution to stain our sample. What is that heavy metal staining solution? Generally uranyl acetate or uranyl formate we are using. So, here you have a list of negative staining solution. In this list of negative staining solution, if I just go, so there is a different type of staining solution you can use it, but mostly we are using uranyl acetate and uranyl formate, because these two staining solution is a very good staining solution and widely used, and it is well established. Otherwise, you can use sodium, potassium, phosphotungstate or ammonium molybdenate. Ammonium molybdenate is also used very frequently, and there are some other solution you can also use. So, sometimes some heavy metal staining solution, so something like osmium tetroxide or lanthanide salt, we can also use it to stain the biological sample, but majorly we are using uranyl acetate and uranyl formate. How do we make those solution? Mostly we are using 1% to 2% water by volume aqueous solution. So, whatever will be the volume, based on that you have to wait, and always remember that uranyl acetate and uranyl formate, whenever the staining solution will prepare, we have to adjust the pH there. So, we have to adjust the pH, and then there will be uranyl acetate or uranyl formate crystal will be also there. So, that is the reason it will be better to filter these two solution before you use it. So, first you have to weigh the uranyl acetate or uranyl formate, and then you have to dissolve that one. Sometime we need a hot water to dissolve that, and then we have to adjust the pH, and then we have to filter it, and when it is filtered, that solution will be ready. Most of the time uranyl acetate stay for longer time, but uranyl formate will dissociate very frequently. So, if you have a uranyl formate solution, it will be better to use it within 1 or 2 days and then prepare a fresh solution, or you can keep it uranyl formate, you can keep it at fridge for next couple of days, and before use it you should filter and then use it. But whenever you will filter, so your solution uranyl formate, it will precipitate, so your concentration will be not 2% anymore or 1% anymore. So, but most of the time uranyl acetate is bit stable, you can use it for longer time, but it will be better again to before use, if you are keeping it for longer time, before use it you should filter it and then use it. But so this is if you want to use this negative staining, it is always better to use a fresh solution. Don't keep your negative staining solution for 6 months and then try to use it. Now, here I will show you that how this negative staining solution look like. So, here this is a uranyl formate or freshly prepared uranyl formate solution, and it will be transparent without sign of precipitation. So, here this tells you that this is the clear solution. It does not matter if you are using uranyl acetate or uranyl formate, your solution should be transparent like that, and it is a greenish color. And if it is you are keeping it for 1-2 weeks, you can see some kind of cloudy nature. So, it is not as transparent as this one. So, if you filter it, probably this precipitate will go away, you can get this transparent solution, but the concentration will be not same. So, if you have this kind of solution, it is better not to use this kind of solution, and the uranyl acetate also similar form, you can see it. So, all the time you have to make sure that your grid your solution is very clean, and you are using a fresh staining solution to do the negative staining. Now, here you can use, if I just go here, you can see that in this particular case we are using a negative staining solution, we are using this curved tweezer to hold the grid, and this is the pipette which we can use it to add the sample, and then we can use the another pipette to couple of droplets of the negative staining solution. This black is the droplet of this negative staining solution. Now there is a water droplet, and you can keep your sample droplet here, or you can add the sample directly here. If you just keeping the sample droplets here, you need a good amount sample and you need a good volume. Most of the time we are using only 20 microliter sample for 20 microliter sample and 0.1 mg per ml for negative staining. So if we just making that 3 or 4 different droplet of the sample, you need a huge amount sample and volume will be much higher. So in this, if you have enough sample, you can follow this protocol, or if you do not have enough sample, what you can do, you just use only 3.5 microliter sample and put it on top of the grid. So and then incubate for 30 seconds to 40 seconds based on your sample, how it is going to attach, concentration, glow discharge parameter. You have to optimize your incubation time also. When grid sample is incubated, then excess solution, because you use 3.5 microliter and 3.5 microliter is a very big amount sample here. So the excess solution you have to blot using this filter paper. So whenever you will use this filter paper, you just use the tip of this filter paper and touch at the edge. So this tip of the filter paper should not touch anywhere of the grid. So it will just touch at the edge of the grid and blot the excess buffer and the sample. When it is done, then we can use this water droplet to wash your grid. So this is the step. If you want, you can skip. In many cases we do not want to wash our sample with water, because if we wash our sample with water, the buffer means whatever salt or glycerol, it will just diluted drastically, and then protein may fall apart and it will dissociate. That is a problem. That is the reason many cases we do not wash. So you can skip this and you can directly go to this 3 droplet staining solution. You stain here, use this grid, and rotate this grid, this particular surface of this grid on top of this solution for some 10 seconds, 15 seconds, and do the same thing in this droplet and this droplet, and finally blotted the excess staining solution using this filter paper. Now sometime for the membrane protein or some kind of protein, you have a high salt or high detergent. So you have this water droplet to wash this grid. So you add the sample here, then use this water droplet quickly to dip this grid, this surface of the grid, touch this water droplet 3 times, and then blot this water, excess water, and then using this staining solution and stain it quickly. Now based on your sample, you have to modify this method. So modify this technique, and in the practical session or the laboratory part, you can follow our videos and you can see it how we are preparing this negative staining grids. So there will show you what is the actual procedure to do this. So we will do these things for membrane protein with water droplet, without water droplet. So there it will be much more clear. Now here also you can see that when you do not have the water droplet, what we have, you have a freshly prepared uranyl format. So this freshly prepared uranyl format is here. So you are using this uranyl format solution and trying to stain it, and then excess buffer you are just from the side of the grid, you are trying to blot it away the excess stain. So do not keep or wait for long time. So your staining solution will deposit on top of your grid, that is going to damage your grid surface, and otherwise it will be too thick stain and you will be not able to see your sample properly. That is the reason this blotting, how you are doing this blotting, it is very important. And also when you have added your sample here and After incubation period you should blot the excess buffer solution, otherwise this excess buffer solution, if you touch the staining solution immediately, your staining the percentage of the stain or the strength of the stain will diluted. That is the reason in this particular step you blot the excess buffer immediately, you use this staining solution, stain it, you can stain for 10 seconds, 20 seconds or 30 seconds based on your sample, based on your grid, surface property of the grid, glow discharge, how you did the glow discharge, you have to optimize this staining procedure, and it also vary based on your buffer solution or what kind of sample you are imaging. So, now when you did that negative staining, so this slide is taken from this slide is taken from the sample preparation and optimization slides. So, here you can see this is a very nicely described in this particular slide how we can do that negative staining. So, here you have a grid and then you are adding the sample. So, you have a filter paper, that triangular shape filter paper, you are just blotting that excess buffer solution. When you blotted that excess buffer solution, there is no buffer anymore, and also you should not wait for long time here, because if you wait for long time your sample will be completely dried and it will damage the sample as well as it will damage the carbon layer also. So, immediately you use staining solution, stain it and blot the excess buffer excess staining solution, and then you will get the final grid. So, this is the negative staining procedure. Whenever you will start this entire process, it should not take more than 5 minutes. Now, what actually happening? What is the principle behind that? So, you have a support film. That support film means you have a plastic coating, then you have a carbon layer. Now, you have a sample, and then sample, when you are using that sample you blotted, your sample will attach with that carbon layer. And then when you are using that staining solution, stain will coat the background and your sample will stay here. And when the electron beam will pass through this one, when electron beam is going through this sample, what will happen? Where there is a heavy metal staining solution, if you remember that the scattering, what is the scattering part? When there is a nucleus, there backscatter is happening, and these are the orbital when electrons are there, and some cases electron will displaced and some cases it will scattered like that. It will be very close to the electron and it will scattered. That time, during this diagram I also told that for biological science sample it will be only hydrogen, carbon, hydrogen, nitrogen, oxygen, sulfur and phosphate, sometimes some metal ion and very few. So, scattering will be very less, most of the time it will just go straight like that. But when we are using uranyl formate or uranyl acetate, uranium is there, which is 92. It is a very heavy atom. This very heavy atom, it will deposit everywhere. It is depositing here. So, here the scattering will be much more stronger than the scattering here. So, in this particular sample the electron beam will not scatter that much, but where the scattering will be much stronger where there is a thick uranyl acetate. So, what will happen? Your background will appear as a black and you can see your white protein molecules. This is the example where you can see the background area, it is a black area, and then this is your protein sample, this is your protein sample. But if you look at the surroundings area, there is a blackish impression is there. That means your heavy metal stain solution is deposited in this nearby area and your protein is appearing as white. So, for negative staining you are using uranyl acetate or any heavy metal, that is the reason scattering is stronger and the background is appearing as black. So, in this entire area you can follow and you can see background is appearing as black and your protein is appearing as white. Same thing here also, this patch this entire patch. Here also you can follow the same thing, that okay, there is a black patch and the protein is appearing as white. This will be exactly opposite when you will do cryo-EM study. So, your protein sample will appear completely differently. So, this is why we are using heavy metal staining solution to stain our sample. Now, there are couple of drawbacks are there that we have to discuss for that negative staining. Now, what we discussed that okay, we have a grid, and then this is my sample, last one we draw it like that, and now this is my staining solution, it will surrounded the particle. Now, the same diagram we are drawing it in a different way. There are three different possibilities are there. So, there is a carbon layer. Okay. So, now I have a staining thickness. So, how much stain is deposited on top of the carbon coated grid, because I also mentioned that is very important, that how we are blotting. If we do not blot it properly and if entire staining solution is deposited, you will get a very thick stain and you cannot see your sample properly. So, that is also not a good situation. That is the reason blotting, concentration of the staining solution, how much stain it is absorbing your sample, all these parameters are very important. So, that is the reason we need to practice these things properly. So, now if we just consider that okay, so this is basically my sample, we just consider that this is my sample, which is attached to the carbon layer. So, same thing here also, which is attached here and here. Attached here. Now, I am using a staining solution. That staining solution stain that sample in only this much is deposited. So, we give the staining solution, you did not wait too much and immediately we blot it. So, what happened, whatever is the staining thickness is very low if you compare with our protein sample. So, the protein sample or biological sample did not embed it properly in the staining solution. Other possibility is that the staining has very nicely distributed like that. So, its thickness is appropriate with our sample. So, in this case it cannot distinguish between the background and protein sample, because scattering will be not that strong here. But in this particular case you have a better thickness. What will happen here, the scattering will be very strong and you will get a very nicely distributed images, what I showed last time. The other possibility is that you have a very thick stain. So, your stain is completely saturating this. So, this is very thick stain and it is covering your sample also, something like not like this less amount stain surrounding outside of your protein. So, if the thickness of your sample if let me just make it little bit better way, so it will be easier to understand. So, if you just consider that okay, your staining thickness is like that. So, what is happening here, the scattering will be very strong, here also it is deposited too much stain. So, in all the cases scattering will be very strong, it does not matter protein is there or not, your scattering will be very strong. So, then this is basically light stain. This will be proper stain. This is called thick stain. So, I will show you these three different example, that time it will be clear that what is called the proper staining or thick staining or light staining images. So, that is very important, we should know how to make our sample properly. Okay. Now, we already covered those parts and we will go for the next one, that that is called the metal shadowing in the next class. That next class we will talk about the metal shadowing, how we can do the metal shadow, and that is also very important. And we also have to talk about the carbon sandwich method, and previously in 1980, 1990 those techniques are very popular, how to do the carbon sandwich method as well as the metal shadowing for the DNA. And then we will come back and discuss about the sample preparation part, or what is the application, good data, bad data, and the application of the negative staining methods. Thank you all for your attention.